Hello all, respected ma'am and dear friends. Today I am going to take my seminar on the topic the Operational Taxonomic Unit. Operational Taxonomic Unit or OTU comes under numerical taxonomy which is the similarity of an organisms and to classify them based on the affinity. And along with those affinities, certain external features has also been taken for classification. Those attributes are known as OTU or Operational Taxonomic Unit. They can either be biochemical, cytological, ecological or developmental aspect. So let's discuss about Operational Taxonomic Unit or OTU. Operational Taxonomic Unit is an operational definition used to classify groups of closely related individuals. The term was first introduced in 1963 by Robert R. Sokal and Peter H. So OTU is a classification of organisms which are closely related. It was first found by Robert R. Sokal and Peter H. Smith, in his contest of numerical taxonomy, he said that an operational taxonomic unit is simply a group of organisms that currently being studied. That is the definition that was given by Smith in his numerical taxonomy. In this sense, an OTU is a pragmatic definition to group individuals by similarity equivalent to but not necessarily in line with classical Linnaean taxonomy or modern evolutionary taxonomy. Nowadays, however, the term OTU is also used in different contexts and refer to cluster of unknown or uncultivated organisms grouped by DNA sequence similarity of a specific taxonomic marker gene, originally coined as MOTU or Molecular Operational Taxonomic Unit, that we will discuss later. In other words, OTUs are pragmatic proxies for species, microbial metazoan at different taxonomic level. In the absence of traditional system of biological classification, as they are available for macroscopic organisms, OTUs are been important factor for the microscopic taxonomic levels. For several years, OTU has been most common used unit of diversity, especially when analyzing small subunits like 16S prokaryotes or 18S rRNA eukaryotes, marker genes, sequence datasets. Operational taxonomic unit has again been divided into four. First one is binary character, second one is qualitative, third quantitative character, and at last the fourth one is data processing. Binary character possesses contrasting state presence or absence of certain features. So certain features, whether they are present or absent, based on these two attributes, organism has been classified. Second one is qualitative character. Based on certain quality of that organisms, they have been classified. Such as if a flower character is taken, whether they are yellow or white, less similar, dissimilar entities or more similar, certain entities has been taken. Third one is quantitative character, from where the quantitative affinities or quantitative attributes has been taken on amount of chemical produced by bacterial strain or the length of the spore. Certain quantitative characters has been taken into consideration. And at last, that is the data processing, where with the help of digital media, data has been entered on a computer and their similarities and dissimilarities has been studied. On the basis of similarity between OTU classified by character. So here you can see there will be number of chosen characters, and from that character certain attributes will be present or certain attributes are similar and certain attributes will be absent. Certain attributes will be, there will be no comparison with the entry that you have given. In that way, you can classify organisms. So, four types of operation taxonomy has been given. First one is binary, second one is qualitative, third quantitative, and at last data processing. Molecular Operation Taxonomic Unit or MOTU. That's the next topic. In MOTU, it is by sequencing an informatic segment of DNA, it is possible to define MOTU or Molecular Operation Taxonomic Unit. So mainly a segment of DNA, informative segment of DNA is taken and it has been sequenced to find out their characters or their attributes, and that procedure is known as Molecular Operation Taxonomic Unit. To be useful, a segment of DNA must be known to the orthologous between species. A parallel will define gene rather than organismal tree. A particular gene is mainly considered, and the segment must encompass sufficient variability to allow discrimination between MOTU useful to research program or defined in other, for example, biological or morphological way. In the MOTU concept, a taxa can be identified through sequence identity. A DNA segment or information DNA segment sequence ആയിരിക്കും നമ്മൾ എടുക്കുന്നത് ഒരു MOTUവിൽ നോക്കുമ്പോഴേക്കും. We are not taking the morphological or any other biological aspect. We are taking a particular informative segment of DNA and that has been sequenced. So that sequence identity is mainly taken into consideration. Identity in sequence need not correspond to identity of OTU as measured by other models. OTU and MOTU are two different things. So OTU is mainly measures based on their biological and morphological characters, but MOTU is mainly on the sequencing of DNA segments. Identity in sequence could mean the same taxon or there is insufficient variation in the distinct taxa. The same operational problem plagues other biological or morphological method of defining. So, if two sequence has been they are identical, then that means they are from the same, either they are from the same taxa or there is some insufficient variation to define distinct taxas. Some the variations are not that much different. That way we can't distinguish between two taxa. Okay. The difference in sequence between specimens can arise in two ways. So the difference can occur mainly in two ways. First is that the difference as a part of natural. It can be naturally occurring difference or within the OTU variations, morphologically variations, other example, or are due to sequencing errors, some errors that has been occurred when the sequencing has happened. So that is the first method where it can either be natural or certain OTU variations can occur or it can be due to the sequencing error. Second is that difference are related to distance between taxa. When the two taxas has been from different organs or from different taxas, then there will be a difference. It is thus necessary, as with other methods or biological or morphological, to use heuristics for MOTU distinction based on known error rates in the measurement and perceived level of difference at that distinguished useful OTU. So the errors should be eliminated during MOTU. For MOTU, these measures can be made explicit. For example, for a non-accepted taxa within a particular group, the level between taxa within group variation can be measured. And the second one is multiple resequencing can also lead to an observational error rate. The comparison between the taxon, the difference rate and within the taxon variation and error rate will define the accuracy and specificity of the MOTU measurement. Given that, it is clear from many gene sequencing that different higher taxonomy groups can differ markedly in their background and adaptive substitute rates, and that different sized population might be expected to harbor different level of within taxon variation. It may be necessary to define different heuristics for MOTU designation depending on their higher taxon study. Now, what are the benefits of MOTU? The data can be obtained from single specimen, often without comparison, parallel or subsequent morphological identification. Second one, the morphological indistinguishable taxa can be separated without the need of live material. So, a live materials use is not necessary. Third one is a single technique is applicable for all taxa. That's one step on taking the DNA and sequencing it. From that single step, we can get information about the taxa. Thus, a long and partial training in particular groups is not necessary. Group or subgroup, it is not necessary. Resource can be made to publish monographs, keys to understand the non-biological properties of the identified MOTU and their close relatives. All stages or morphs of taxas are amenable to the study as the method depends on genotype, not phenotype. In addition, the MOTU data, the sequence are suited to exhaustive and model-driven phylogenetical analysis to derive independent and testable hypothesis of OTU interrelatedness. MOTUs are also used in DNA barcoding. In DNA barcoding literature, the designation MOTU has been widely used to describe the cluster of sequence. DNA barcoding literature, MOTU can designate different situations that were interpreted as belonging to three distinct groupings. A group of undefined organisms sharing similar sequence, a group of organisms with a species that are distinct at molecular level from the members of the species, a group of organisms that differ species that are similar at the molecular level. So based on DNA barcoding with the help of MOTU, organism has been divided into three. Now, with the help of MOTU, we can separate organisms that were first considered as a single taxon, but with the help of MOTU, they were separated because with the help of MOTU we can understand that they were two different organisms. For example, the African elephant, Loxodonta africana, was considered as a single species on the basis of their morphological data. But based on their morphological and biologically, they were considered as a single species. But after on classifying them based on their MOTU, they were reclassified as two species with the inclusion of molecular data. So these were the importance of MOTU. Next is integrated taxonomical units or IOTUs. Integrated operation taxonomic unit. The molecular techniques, that are MOTUs, were widely used or widespread tool for the identification of biological entities. However, until a few years ago, their application to taxonomy provoked intense debate between traditional and molecular taxonomists. So there was a conflict between the traditional taxonomist and also the molecular taxonomist. So to standardize these conflict or to standardize taxonomical definition, the IOTUs or integrated operational taxonomic units were introduced. IOTUs comes from the concept of operational taxonomic unit and parallel to the molecular operation taxonomic unit. So they comprised both the OTU and MOTU. The latter is largely used as a standard in many molecular based works, even if not always explicitly formalized. However, while MOTUs are assigned solely on molecular variation criteria, OTUs are identified from pattern of molecular variation that are supported by at least one more taxonomic character. Now, how are IOTU and MOTU are different? Now, IOTUs are more informative than the general concept of OTUs and the more recent MOTU. MOTU, OTU which consider IOTUs are different. They are little more informative and they are little more strong. According to the information content, IOTUs are closer to species, although it is important to underline that IOTUs are not species. MOTUs are assigned solely on molecular variation criteria. IOTUs are identified from pattern of molecular variation that are supported by at least one more taxonomic character. So here taxonomic character is also supported and also the molecular variations. Like MOTUs, IOTU describe organisms that are similar at molecular level, that is they share a DNA barcode. But unlike MOTUs considered in the original definition, they also share at least one other characteristics from the taxonomic circle. IOTUs have higher information content and a strong taxonomic support. Overall, the use of more precise panel of taxonomic entities increases the clarity in the system of field and has potential to fill the gaps between modern and traditional taxonomy. From this figure, we can understand that if we select a number of individuals and we can separate them into different species based on their morphological type. That is called OTU. We can directly separate them into different species based on their morphological type. But while considering IOTU, there it is taking the information from the morphological type as well as from the MOTU or molecular operational taxonomic unit. From the two informations, they are gaining a single strong supportal evidence for the species and they are creating a separate species. So the evidence for separation of species will be or classifying the species will be more stronger in IOTU rather than the morphological symbol OTU or MOTU. So just taking the molecular informations and dividing or classifying organisms, or by taking the morphological informations and classifying organisms, IOTUs is far much better because it is considering both the taxonomic, both the morphological character as well as the molecular character. Next, we are going to deal with another topic, that is Global Taxonomic Initiative or GTA. The GTA emblem is given in the right. Global Taxonomic Initiative or GTA is a cross-cutting program developed by CBD to address the taxonomic impediment. It is a program developed by the Convention of Biological Diversity, CBD, to address the taxonomy for the taxonomic persons or the taxonomist and for the taxonomic programs. This initiative has been taken. It underpins the conservation of biological diversity, an international policy used to inform national policy by the parties to the CBD. The main aspect of GTA is to ensure that the taxonomic information to the taxonomist, decision makers and non-taxonomist. It is mainly for the taxonomist and also for the decision makers and for the non-taxonomist. Darwin Declaration led to the development of GTA by a meeting of group of experts held in Darwin, Australia in February 1988, was sponsored by the CBD, Convention of Biological Diversity (CBD), the GEF, Environmental Australia, and Smithsonian institutions. The program of work POW for GTA focus on supply taxonomical information and work to implement the objective of CBD. It is adopted in 2002, supplemented in 2006 COP decision, and it summarizes objectives and major activities of GTA. It is comprised of 19 planned activities within 5 operational objectives. So there were 5 operational objectives, and under that there were 19 planned activities. One specific target is included in operational objective 3. So there were about 5 operational objectives and 19 planned activities. From that, the first operational objective was assess taxonomical needs and capacities at national, regional and global level for the implement of the convention. So in that, the first planned activity was to country-based taxonomic need to assessment and identification of priorities. Second one, the regional taxonomic needs assessment and identification of their priorities. Third, global taxonomic need assessment. Fourth one, public awareness and education. The second operational objective is to provide focus to help build and maintain the human resources, system and infrastructures needed to obtain, collect and curate the biological specimens that are the basis of taxonomical knowledge. And under the second operational objective, there were 2 planned activities, 5th and 6th. The 5th one was the global and regional capacity building to support access to and generation of taxonomical information. 6th one, strengthening the existing network of regional cooperation in the taxonomy. The third operational objective, which is the important one, it facilitated an improved and effective infrastructure system for the access to taxonomic information with priority on ensuring countries of origin gain access to information concerning elements of their biodiversity. And under that, the 7th planned activity developed a coordinated taxonomical information system. The 4th operational objective within the major thematic work program of the convention include key taxonomic objectives to generate information needed for decision making in conservation and sustainable use of biological diversity and its components. So under that, from 8th to 13th planned activities were under operational objective. The 8th one was forest biological diversity, 9th one marine and coastal biological diversity, 10th dry and subhumid lands biodiversity, 11th inland waters biological diversity, and 12th agriculture and biological diversity, 13th mountain biological diversity, 13B island biological diversity. The 5th operational objective within the work on cross-cutting issues of the convention include key taxonomic objectives to generate information needed for decision making in conservation and sustainable use of biological diversity and its components. So under that, the 14th planned activity, access and benefit sharing, 15th invasive alien species, 16th support in implementation of Article 8, that is traditional knowledge, innovation and practice, 17th planned activity, support for ecosystem approach and CBD work on the assessment including impact assessment, monitoring and indicator. The 18th one, protected areas. Now next is the developing the GTA and monitoring its progress. So GTA has mainly dual nature, that is a policy and implementation. The policy of GTA is to identify the priorities for action which culminate in decisions of the COP including the program of work, and second one is the implementation. Implementation policy at international level into policies, strategies and activities at national level. The taxonomic tools used for GTI includes BioNet. BioNet is an initiative that promotes taxonomy so that countries can identify their biodiversity, address sustainable development and conservation priorities. It supports Global Taxonomic Initiative or GTA. Next is the Biodiversity Information System or BIS. So first one is BioNet and second one is BIS. BIS provide taxonomical information over the architecture for sharing national, regional and global biodiversity databases. Now there are certain examples for the GTA project, including the national project, a partnership for enhancing expertise in taxonomy, US, GEF project, the Indonesian Biodiversity Collection Project, GF project, a SABONET, Southern African Botany Diversity Network, the GF project Botanical and Zoological Taxonomic Network in Eastern Africa, BOZONet, linking conservation to taxonomy, GF project Biodiversity Resource Development project for Costa Rica, and non-governmental investing in nature and eco partnership between HSBC Group, WWF, Botanic Garden Conservation International and Earthwatch. In 2002, the HSBC Group established in eco partnership with WWF, a Botanical Garden Conservation International, BGCI, and Earthwatch. They investing the nature program 139 with $50 million to fund conservation project around the world over 5 years. So in conclusion, with GTA, we can say that it is a cross-cutting issue of United Nations Convention on Biological Diversity to address the lack of taxonomical information, and there were 5 operational objectives of GTA are of each member's country to conduct a taxonomic need assessment, contribute to capacity building, provide support for the CBD thematic areas and cross-cutting issues, and improve access to information. So these were the references that I took for this seminar. Hope you all understood. Thank you.